Welcome, online listeners and viewers, and thank you for joining the Science AAAS webinar, Interrogating Cancer Drivers via CRISPR, mutant TP53 as a therapeutic target in hypodiploid B-cell acute lymphoblastic leukemia. My name is Jackie Oberst, and I'm the Assistant Editor for Custom Publishing here at Science. I'll be moderating today's webinar. Joining us today as a guest speaker is Dr. Ernesto Diaz-Flores from University of California, San Francisco, who we will learn more about shortly. Before we get started, I'd like to orient our online viewers to what's currently being seen. At the top right of your screen, you'll find a photo of today's speaker and a view bio link, which you can click on to read more details about his background and research. To the right, you'll also see the Resources tab, where you can find additional information about technologies related to today's discussion and a PDF of the slides. After the speaker presentation, we will have a short Q&A session during which we will answer some of the questions submitted by our live online viewers. So start thinking about some questions now and submit them at any time by clicking the Ask a Question tab, also on the right, typing the question into the message box, and then clicking Submit. You can also log into your Facebook, Twitter, or LinkedIn accounts during the webinar to post updates or send tweets about the event. Just click the relevant icon at the bottom left of the screen. For tweets, you can add the hashtag #ScienceWebinar. Finally, thank you to Levitas Bio for sponsoring today's webinar. And now, let's begin. In the last few years, interrogation of the role of altered genes in cancer has been greatly facilitated by the use of CRISPR-Cas. This gene editing approach is a key research tool for modulating gene expression, knockout and knock-in, efficiently and precisely. However, this approach can be limited depending on the cell type, hard to transduce or primary cell. Such limitations are a challenging roadblock to advancing studies of a wide spectrum of malignant cells. Patients with hypodiploid B-cell acute lymphoblastic leukemia present with very poor prognosis, a 5-year event-free survival of under 29%. These cells have been shown to exhibit mutations in P53 in over 90% of cases. To better understand the mechanism of resistance that arises from P53 mutations, we wished to generate isogenic hypodiploid cell lines, including NALM16, deficient in P53, and with knocking alleles of P53 wild type or different mutant versions. This seminar will cover the steps taken to go from 1% to 2% lentiviral transduction efficiency to over 95% in hard to transduce hypodiploid cell lines and primary cells. We will present how we were able to quickly and gently enrich transduced clones to allow such high transduction efficiency and the generation of knockouts for further studies. Without further ado, I'm very pleased to introduce our speaker to you now, Dr. Ernesto Diaz-Flores from University of California, San Francisco. As a leukemia researcher, Dr. Diaz-Flores' ultimate goal is to find new cures for high-risk leukemia through an understanding of the mechanisms mediating leukemogenesis. During his time as assistant professor at the University of California, San Francisco, he has identified B-cell lymphoma 2, BCL2, as a promising therapeutic target and validated venetoclax as an effective drug against this leukemia. These studies contributed to the first clinical trial of venetoclax in pediatric acute lymphoblastic leukemia, or ALL. In follow-up work, Dr. Diaz-Flores provides evidence for a highly effective combinatorial therapy as well as biomarkers or response to inotuzumab immunotherapy. His current work focuses on studying pharmacological and immunotherapeutic opportunities for targeting mutant P53 in the context of acute leukemia. This work represents a new paradigm in the field of P53 and hypodiploid leukemia and is expected to bring novel treatments to the clinic. Welcome, Dr. Diaz-Flores, and thank you for joining us today.
Thank you very much, Jackie, for the great presentation and introduction, and thank you, AAS, for inviting us to present our work. So today I'm going to be talking, as he's been introduced, about how to maximize CRISPR efficiency to interrogate cancer drivers. We studied that in the context of mutant P53 as a therapeutic target in hypodiploid leukemia. I'm an assistant professor at UCSF, and I've been focusing on this high-risk leukemia to try to identify better treatments for it. So hypodiploid B-cell leukemia is characterized for having less than 46 chromosomes. It accounts for 2 to 4% of pediatric ALL, but due to its poor prognosis, it really has drawn more attention in the last few years, particularly our groups and some collaborating groups in St. Jude. There is 4 karyotypic subgroups based on the chromosome content of these leukemias. 44 to 45 are near diploid, 40 to 43 high hypodiploid, and those have a standard risk for standard treatments. But then there is the 32 to 39 chromosomes called low hypodiploid, and 25 to 31 chromosomes near haploid leukemia that has very poor prognosis. And has been recently shown these patients do not benefit from even hematopoietic stem cell transplant or even the most aggressive chemotherapy regimens. And therefore, we need new treatments for this leukemia. So, focusing on the low hapodiploid, the 32 to 39 chromosomes. So this leukemia has a very silent mutational landscape. So it particularly has mutation in p53. So over 90% of patients have it, 40% of them at germline, so representing Liefers-Menis syndrome. Additionally, there is some mutations or deletions in our retinoblastoma, CDKN2A and B. But besides those, there is no many others. So it's a very silent in terms of mutations. There is no fusions. So it also represents a very good way to study these particular mutations, particularly p53. So we did the largest transcriptomic analysis of hypodiploid patients, including 124 hypodiploid patients, and comparing to 7 other subtypes of B-ALL, including Ph positive, Ph like, and CR left, TCF3, PBX, and so on. So with those, we're able to do principal component analysis to see how they distinguish, and we can see on the left all the diploid and near diploid clustered together, the near haploid clustered together, and the low haploid also clustered together but separated from the others. So those, as I mentioned, the patients have like over 90% of them have mutation 53. So we did a network analysis. All the top 1,000 dysregulated genes were connected to p53. Using gene set enrichment analysis, also the pathway is really dysregulated, and we are able to distinguish differentially gene expressed in the p53 mutant leukemias versus the diploid B-ALL. So we consider that hypodiploid leukemia is an ideal model to study p53. The main reason is because most of the patients have this mutation. It has a complete penetrance because there is only one chromosome 17 where p53 is, and therefore the other locus is lost. So there is no the other chromosome with a wild type p53. So all the phenotype is completely penetrant. And then, as I mentioned, there is low mutational burden, so there is not many other mutations that accompany the p53. Like most of the dysregulated genes are connected to this gene, and then we can see differential expression of multiple genes, and you can see here in the inset on the right that the yellow or orange represent the other diploid subtypes, whereas the green represent the low haploid. And you can see how, like using a box plot, how the genes are differentially expressed, very clearly seen. So with that in mind, we know that there is multiple different mutations in p53, even in the hypodiploid subtype. We have looked at the protein expression, and we see that most of the mutations accumulate. And when they accumulate, they can acquire gain of function. So there are gain of function mutations that can acquire oncogenic properties. And then we see that it's missense, frame shift, and nonsense mutation. And what we think is what makes the difference is not just the type of mutation, but the length of the expressed mutant protein. So as long as there is enough protein, they can have abnormal alterations. And the reason is because p53 is a transcription factor, so it can bind to different DNA binding sites, but also binds multiple other associated proteins to mediate this transcription. And like all these mutants accumulate in an altered conformation, so they can bind new players that were not bound in the wild type conformation. So the workflow that we are using for our studies is we take the low haploid PLL samples from patients, They only have like one chromosome 17, fully penetrant mutation. We divide them based on somatic, like somatic or germline, but also in frame shift, missense, and nonsense, so we can do a systematic study. And then we have 3 hypodiploid cell lines, 2 that were commercially available, NAM16 and MHH-CoL2, but also one that we generated in the lab, BEC1732. So we now can look at the kind of the difference between mutants and do like affinity, proximity, mass spec, and be able to see what are the new binding partners that happens in the mutant context. So for that, we will first knock out 53, whether wild type or mutant, from these hypodiploid cell lines, and then reintroduce wild type and then systematically the different mutations. So in that way, we can do an analysis of the different subtypes. So for that, we thought like, yeah, we're gonna do CRISPR in these cell lines and then we just reintroduce, and that everyone's doing CRISPR, so it should be like quite easy. So we started 2 years ago, 2.5 years ago, and we had been very unsuccessful in getting a full kind of like a complete knockout in these cell lines. We knew that they were hard to transduce, but then here I'm basically introducing you to our journey that went from like almost not getting any like transduction 53 and not being able to do CRISPR to the point that we are right now. So we decided to optimize CRISPR from step 1, basically. So we look at the entire workflow: plasmid selection, packaging the cells for transfection, producing the lentivirus, transducing the cells, selecting them, and validating them. So for plasmid selection, we have the CRISPR plasmid that I'm not showing here, but we decided to use these to optimize the CRISPR workflow. So this is a YFP luciferase. It provides us the luciferase so we can do in vivo tracking as well as ex vivo or in cell lines, and use luminescence, which is very sensitive, to do any type of analysis, as well as YFP that we can do analysis by flow cytometry. So for the transfection, we decided to compare the 2 major approaches. One is liposomal based, and the other is calcium chloride. So then we just try to see which method is the best and then follow up the optimization. So for liposomal, we look at what's out there. Most people use, and we have used in the past, lipofectamine, different type of lipofectamine. But more recently, we have been looking into transit LT1 from Mirus. And this graph just shows the transduction efficiency, basically the luciferase expression, based on the different reagents. So transit has a better transfection efficiency in this case than the lipofectamine 2000 and the LTX plus and Fugene, and also lower cell death, because instead of using liposomes, it uses cationic lipids, so that's gentler for the cells. So that's why we decided to use transit LT1. We did some comparison with lipofectamine, and yes, transit was superior. So then, for the liposomal or cationic lipid version, we use Mirus, and where we played first in 6-4 plates, the hex cells to produce the lentivirus. And then we basically mix the reagents like as per the protocol. And then we put different ratio of Mirus to DNA. So the content of plasmid we quantify, and then we put like 2 times Mirus, or 3 times, or 6 times, or up to 12 times. The brochure recommends between 2 and 6 times per DNA content. We went all the way up to 12 versus 1. And we did the same for calcium chloride, same ratio. So when we compare, so I'm not showing here, but the calcium chloride didn't even score like in luciferase. So basically, it was very superior the Mirus, and also calcium chloride, as we increase it, it induced more cell death. So here I'm showing the MARUS results. As I said, like if we overlay this with the calcium chloride results, you would not see still the calcium chloride. So that was the difference. So that, okay, make us focus on the MARUS. And in terms of what ratio to use, so we went from 1 ratio of MARUS versus 2 ratio of DNA to like 6 times the MARUS versus 1 time of DNA. And you see like 3 times, 3 to 1 gave us superior results. So then we also look at the DNA content. So when we say like, okay, 3 proportions of MARUS versus 1 proportion of DNA is accounting for the 3 plasmids that we have put in there. So to generate lentivirus, we use a second generation lentivirus way, which is using 3 plasmids, the envelope plasmid, packaging, and the transfer plasmid. And you can put in different proportions as well. So here we compare what is the ratio between those plasmids, 2 to 2, or do we put 2 to 8, so putting more of the transfer plasmid. Your plasmids of interest in this case would be the CRISPR. For our particular example is the YFP luciferase. So should we put just more of that, or should we put more of the envelope as well as the transfer, or so on? So we did multiple different comparisons, and then this was the results. So basically the 2, 6, 8 was the best proportion. So 2 being, if you go back, 2 of the envelope, 6 of the packaging plasmid, and 8 of the transfer plasmid of your interest. I have shown that 3 to 1 ratio of MARUS versus plasmid work better, and you can see that yes, so if you compare 2 to 1 ratio on the left, 3 to 1 ratio on the right, that was the superior one, and 2, 6, 8 in terms of the plasmid ratio. So now we have the optimized conditions for the transfection of the cells to produce lentivirus. So then comes to harvesting the virus. So we normally collect, so we have like the 100 millimeter Petri dishes to grow the hex cells, and then we put them in 30 milliliters of culture media, and then we culture that, or we harvest that media, and then we concentrate the virus. So we have to compare non-concentrated versus concentrated. So for concentration, we use Lenti-X, Lenti-X concentrator. I believe it's from Takara. So then all our 30 milliliters can be concentrated to 600 microliters. So that allows us to play with volumes better than with 30 milliliters that we wouldn't be able to like add as much in any transduction experiment. So then we wanted to quantify how much virus do we have in those 30 milliliters. And so we found that also Takara was having these gold sticks that look like, I would have said before, like it looks like a pregnancy test, but now I would say like it looks like an antigen test, because I think we're more familiar now with those. So then you can see that once you have the virus, you get the extra band, and then it has an app where you can quantify that and give you like an infection unit number. So the system is very, very simple. So you just take your the volume that you want to quantify, 20 microliters in our case of the concentrated. You add it to these cartridges, add the solution, and then you wait for like 10 minutes. You see the band appear, quantify with the app, and it gives you like infectious unit values. So as you can see here, we started doing that for different production of virus that we did in different days, or same day but different virus. So we got a value of 750 approximately, or 700 infectious units per milliliter. Okay, is that good? It's not good, like no idea, but that's how we are starting. So then we did additional times, additional days, and then we got to one day that we got like 5,000. So that sounds really good compared to the 700. So and we decided to work with that. Looks good, the amount of virus that we're producing. One interesting thing is if you leave the cells or the lentivirus, concentrated virus, at 4 degrees, it decays. And that's something that is being known, but how much it decays, we didn't know, so we wanted to validate. So we saw that over a week, we basically lose like 80% of the viral infectivity, basically. So the viral load. So because of that, we immediately, when we generate the concentrated virus, we freeze it at -80. And we thought in the moment that we want to do for any analysis. And we have calculated the concentration, and it's the same as the day that we froze it. So that works really well. So now we wanted to compare. Okay, we have this virus that we know is highly concentrated. We want to see how to transduce the cells, and then what is going to give us the best transduction, like what in the protocol will make the biggest difference. So the most people, it's either like people use nucleofection, which is using electroporation, or lentivirus production. In our case, we're just focusing for this particular presentation in the lentivirus production. For that, you normally use polybrine to help the virus bind the cells. So we try different concentrations of polybrine. Polybrine is also toxic, so the more you put, the better transduction efficiency will be, but also the more toxic for the cells, so you will kill more. So it's a trade-off. We also try just virus without polybrine, increasing concentration, and then virus at a given concentration, given a volume, 25 microliters from the 600 concentrated, and adding lentiboost. So lentiboost, prostaglandin E2, and cyclosporin H, those came from a recent paper from St. Jude that they did a comparison what works best for transducing cells, lentivirus production, and also allowing that to be translated into engraftment of transduced cells into mice. So because of that, we got a lentiboost, PGE, and cyclosporin H, and we compare which one works best. So here is the results. So in the luminescence that of this transduction in basically like easy to transduce cells, 3T3. So this is our control or reference cell line. So you see as we increase polybrine, high concentration, the transduction works better, not dramatically better, but better. So if we just use the virus, the more you add the 50 microliters over 250 microliters of final culture volume of these cells was the best. And then if we use 25 microliters of the virus, this one, and then you add lentiboost, you greatly enhance the transduction efficiency in these cells. Prostaglandin E2 and cyclosporin H did increase marginally, but not to the level of lentiboost. So then we increase our conditions. We wanted to try different things. In addition to polybrine, transduce it, which is another reagent to bind virus. So also different concentration of lentiboost, a different concentration of virus. And then basically here what you see is the first one is just lentivirus by itself. And let me say, red line here is viability. I just wanted to make sure that whatever I'm adding is not really affecting viability. And then the blue bars is the luminescence, basically transduction efficiency. So for a given volume, 12.5 microliters over 250 microliters of virus, if we would transduce it, it increased, but marginally. Same for polybrine. So, I mean, it helps over not having it, but not that much. So when we add lentiboost, we have a great increase. As you see that this goes from the 600 values to 4,000 and 5,000 luminescent units. So prostaglandin didn't help, cyclosporin didn't help, and then the combination of all three gave us very high, but it's particularly based or driven by the lenti boost. So as you can see here, the levels are 5,000 relative luminescent units in these reference cells, which is great. So now we go back to our hypodiploid cells, hard to transduce, and let's see how the same conditions at the same time, the same day, I mean, it was in parallel. I was doing this, so like same person, nothing changes, and then we got these results, but the level, instead of being 5,000, is 200 relative luminescent units. So really low, really low. If we were like to put this in the other slide here, basically you would not be able to see above the red line here. So not even close. So still very, very low. And lenti boost was the only thing that was really increasing, and also the virus volume. So what happened after that was I wanted to see not only producing the virus the way we are doing, which is like we prepare the hex cells on Monday, we transfect them on Tuesday, and we collect at 48 hours. I think it's the most common, 48 hours. Some people do at 24, some 48, some 72. Some do at 24 and combine with the 48 fraction, and some are the 72. So, and then cells are discarded. So instead of that, I wanted to see what if we add more media to the cells, to the hex cells that are producing the lentivirus, and we let them produce more virus. Is this something that will keep producing virus or not? I just wanted to see, can we keep using the cells, or do we have to transfect them every time or like every week that we want to use this? So we had added more media. So we had collected all the 30 milliliters. We added again 30 milliliters. And we added the media we had collected at 48 hours. So then we added the 30 milliliters on Thursday, and we collected on Tuesday. So these cells have already been producing, and they are not probably dividing anymore because being very confluent by that time. So maybe it's not going to work. Well, what happened is that instead of 5,000 infective units, we got 41,000. I was like, wow, this really made a huge difference. And I had 2 plates, and then the second plate the next day, it was 45,000. So then I took the same cells that I just showed, the hard to transduce, nano-16, that we had been only getting like 1-2%, maximum 5%. We have tried for like months, and I just used this virus that I was like smoking hot, and then in 72 hours, I had like 94%. So I went from like 1%, 2%, 5% to 94%. So the main difference, what makes here, is the virus production. And also, if I collect the transduced cells or I measure them at 24 hours, it was only 17%, 48 hours about like close to 50%, and 72 hours is when we got like the 94%. So again, tell us that leaving the virus there for additional time works. So we also were able to like quantify this virus, which is great because now we know when we get really high levels of virus, then we can try this and we can get the very high transduction efficiency. So from this point on, it's a very different prospect that we are seeing now with CRISPR. So what I did also with this virus is, okay, now we have the patient samples that we want to transduce, and then can I also get high levels on the end? The problem we have with primary cells, and this is overall problem for anyone in the world trying to culture primary patient samples, is that they die over time. So particularly ALL, BALL, there is no real protocol for growing them in culture. There's protocol for AML, there's protocol for TLL, that you can survive adding like some cytokines. BALL do not grow better with any particular cytokine. So, and then you go from like, let's say you start on Monday with like 95% viability. By Friday, you have 5% viability, so almost everything is dead. So you cannot culture them for too long. So then, as I show, the better transduction is at 72 hours and maybe even more, but we cannot take that risk. So we kind of went with a trade-off. So we put them in culture on Monday, and then 48 hours later, we have kind of like half of them are dead and half of them are alive, and they have been transduced. So how do we purify these hide cells? So if we put in a conventional FACS sorter, the shear forces will also increase additional cell death. So the remaining cells that we have from the primary samples are going to be further damaged. If we try to isolate it with like some of the beads that discard the dead cells, the yield is really low because there is a lot of unspecific binding. So this is when we start like highly utilizing the Levy-cell technology. So for those of you who are not familiar with the Levy-cell, is a gentle sorter based on magnetic levitation. So you use a magnetic reagent that basically is liquid support that creates different gradients and separate the cells based on density. So you put the cells to separate. So then in the machine, it takes like 5 minutes. And then the live cells will float, will levitate, whereas all the debris and any dead cells will go to the lower fraction. So then you set up where you want to your line to divide, and okay, I want to take just the live cells, and you go like with the dividing line as close as the live cells as you want. And then you get the upper fraction where you have mostly the live cells. And you can go as stringent as you want, or if you want a little bit more yield, you can be a little bit less stringent. And then if you look at the lower fraction, that's the waste fraction. Basically, that's where you discarded almost all the dead cells. So then having the upper fraction, now we can isolate them, and then we could see that we had like a 15% transduction efficiency. I'm not showing here. The next day, those cells were at 40, something percent, and then and because they are highly viable. So because of that, also like being cells highly viable, we can do the engraftment in mice. We normally use 1 to 2 million cells per mouse to engraft, but because these are highly pure and then also the transduced, I use only 100,000 cells per mouse. And then we got a complete engrafting of the mice. So this has allowed us to culture primary cells, just select the live cells with no additional damage or decrease in viability, and then highly purify the viable cells, and then be able to transduce at way lower, 10-fold, 20-fold lower amounts to do engraftments in mice. So now that we have this, again, this virus producing really high levels, then we decided to see how much volume do we need, and we saw that 100 microliters in a 250 microliters sterile volume gave us 90% in these hard to transduce cells. Fabulous. So then it's like, okay, if we go by 40% volume, we get like 90%. So then we go 40% volume in these 3T3 cells, which we're not using anymore the YFP luciferase. Now we go to the CRISPR plasmid for P53, and we use one for guide RNA 1, guide RNA 2, guide RNA 3, and yes, in the reference cells, easy to transduce, we got like almost 100% transduction efficiency, which is fabulous. But when we look at the non-cysteine, the hard to transduce, now that we moved from a like YFP luciferase, which is an 8.4 kb, 8.8 kb, to a 14 kb, like much bigger plasmid, now with a 40% volume, there is in some of the guide, like some of the plasmid, we only got like 17% or 13%. So then using viral volume from a concentrated stock is not reliable parameter to know what your transduction efficiency is going to be. So we need to quantify. And as you see, like different batches can give you different virus. You plate them at the same time, use different plasmids because have different guide RNAs, and you get different viral titer. So you cannot assume that because you are putting the same, everything plating the same way, you're gonna produce virus in the same way. So you need to be quantifying each of them and adjusting appropriately. So we need to quantify, so then we order more of the Go sticks from Takara because we have run out of the one that we had. But of course, we are in times of COVID and supply chain has been a problem. So it was back ordered for 6 weeks. And I had plated like multiple different plasmids for CRISPR that week, and we could not wait. It ended up being more than 6 weeks actually till we got them. So immediately I ordered ELISA kits. They are more hands-on, but we need to quantify. So we use different ones. We do side-by-side comparison, at least 2. So, and then one, it kind of helps you purify. I mean, I use Expert Bio and Cell Biolabs, but you could use different ones and just try it. And then here, what you can see is the color. So this is in pink where we have the cells and the strips. And the next one is the blue. So the first 2 we're doing in duplicate. The first 2 strips in vertical are the standards from the highest concentration of like P24 protein to the lowest. You see like the light blue to like white or translucent. The next 3 are arrovirus, like really, really blue. So like off the chart. So when we quantify that, we saw that first, using the standard curve, there's not a real good linear regression. So this is using absorbance. Absorbance has very low dynamic range. So if we want to have a good linear regression here, we have to only focus on the like intermediate concentrations. And that's where you get the linearity close to 1. The coefficient of variation is like R 0.99. We only had to be within these 3 concentrations. So that tells us where the linearity of the absorbance reader is. So now with here's the standards and how we go from the highest concentration to the lowest, and then our virus. We also went like 1 in 10 dilution, 1 in 100, 1 in 1,000, 10,000, 1 in 100,000 dilution because we knew that the virus was really high. In a blue gradient, I put the concentrations where the absorbance is linear. So you want that your virus, when it's red, at whichever dilution, it falls within that linear range. So that linear range in our case was when we use a 1 in 10,000 dilution of the virus. So that gives us the value. So then you just multiply by 10,000, whatever you get. And then we got that instead of the 200 nanogram per milliliter of the highest standard that we had from the kit, our cells had like 400,000 nanograms per milliliter. So we are really generating a lot of virus here. So now that we have those, we know we can calculate the transduction units and also the multiplicity of infection. So basically, that's where like this, be able to quantify the virus, give us numbers. So then, you know, like the transduction units that you have, and for 1 in 1,000 dilution, you got 24% of GFP. So then, based on that, you made the calculation of MOI, meaning MOI, for instance, of 1. How much volume of virus do you need to have 1 viral particle per 1 cell? So it tells us 9 microliters. Okay, so this is with a YFP plasmid. I use YFP in high concentration of cells. Just wanted to make sure that the cells that I'm using to transduce is not changing, so that the MOI doesn't change based on how much of those cells I'm adding. So if I'm adding 3T3 at 100,000 or 500,000, I'm gonna get the same readout. So in orange shows also the MOI for 1 in the linear scale. So basically 10 and 9. So that's really, really consistent. But now, if instead of the YFP, if I calculate for the P53, it tells us that it's 345 microliters that I'm gonna need in order to get like the 100%, right? So now you have, so that would be like for 1 million cells. So you can like scale it down. So 34 microliters for like 100,000 cells. So we did that in non-cysteine, and it was the same. So for the YFP, 8 or 5 microliters, so basically like let's say 7 microliters to get 100% from this YFP luciferase virus that we have produced in such high levels. The P53 CRISPR plasmid, we needed a little bit more, right? 2,000. So that would be for 1 million cells. So then if I plate 100,000 cells, I only gonna need 200 microliters, but that's still in a 250 microliters. So then I can go like 50,000 cells. So then 110 microliters of virus still give me extra volume up to 250 of my media. So then I can get what I want. You can go for an MOI of 0.5 instead. So that's showing how we have very good. But again, we have multiple different CRISPR constructs that we are now using. Now for like CDK9 or MCL1 or BCL2, whichever your gene of interest is. And then you transduce the cells, and then in this case, we're using 100 microliters of virus. I probably should have put this slide prior in terms of the volume, right? So we're considering, okay, 100 microliters of virus will give us the same amount than 100 microliters of the other. But no, in this case, we have very little GFP, like 1 to 2%. But then with this, now we can calculate the MOI. So for an MOI of 1, we'll need 4,000 microliters of this virus. So then again, that's for 1 million. So then you scale in 100,000 for 100. So in 10,000, 47 microliters. So now you know how to then like see the cells and how much virus to get to the 100% transduction efficiency or 50%. So in that way you can have a little bit more cells. So in that way we went for a MOI of 0.5. So we're aiming to get like 50% with the different ones in these hard to transduce cells with all the different new plasmids that we have calculated. And for most of them, you can see that they are like close to the 50%, 40, 38, 40, 39, 50, 43, 39, 45, and so on. So now like we can now CRISPR 53. That was the goal of doing all this. So we generated the different guides against exon 1, 3 different guides. And then we were using 100,000 cells. And then from the 100,000 cells, that is very little amount still, but then we want to have like the MOI, like high MOI. So then again, we went to the Levy cell. And then what we can do is lentivirus transduction still will kill cells, right? Because of the different reagents that you use. So then in the upper fraction, you see the live cells. So like 60% of cells were alive. This, I believe, is 48 hours. So then we use the Levy cell and we purify all those live cells. And we discard the dead cells. So basically now we have 40,000 cells like alive, but they're pure, highly viable. And now we look at the transduction efficiency of those, and we're aiming like for the 50%, and we got like 60% to like 7%, around 60% to all of them. And then that was at 48 hours. The next day, 72 hours, we go like to 75%, and even for the pool, we got 95%. So, and now we finally went to the validation part. So now we got the knockouts at 95%. Let's see how that looks in terms of the knockout of the gene. And then you can see first NAM16 control cells, they have the mutant 53 that highly accumulates because it cannot be degraded. Non-targeting control looks the same, but the guide RNA1 highly reduces the expression. The second guide didn't work as well. The third guide works much better, and when we did a pool, we had a complete knockout. And then we put URCAT here as a negative control because URCAT doesn't have 53. And then that's by Western. On the right is using qPCR, and you see also the levels in the 53 knockout pool that we completely eliminated the gene. So, and this is in bulk. So we haven't even done at this point the single cell selection to then sort which clone got the best indels, which is a common practice, like people will get like 5% transduction efficiency, we'll sort, then we'll sort for those cells that got CRISPR, and then do single cell sort to pick those that the indel was very effective. So this is in bulk, so still. So the next steps is basically do that in more cells with more different genes of interest. So we're interested in CDK9, MCL1 for like a paper that we're finishing. We got already those results. We have the inducible in vivo CRISPR that we use the Spectre, and then currently we are we have been ramping up in terms of genes because now we can get to like really high transduction efficiency in a very controlled way for any cell type or any construct that we want. And then in terms of the biological aspects of the next steps is the phenotypic characterization. So the doubling time of the knockouts of the NAM16 that has the mutant versus NAM16 knockout with changes, or the gain of function analysis that we can do, drug sensitivity against drugs that we know that they works, but they also can have resistance based on 53. And then now we can finally going to interrogate what the effects of the different mutations of 53 happen. And then we are we have the cell lines, we have the different mutants, and we have introduced the wild type and the different other mutants using the same transduction protocol. Now we have very high levels, and then we purify, and then we are doing the RNA-seq and the affinity purify mass spec to do the quantitative biology analysis on them. So then for the quantitative analysis using affinity purified mass spec, we can see what are the binding partners of 53 in wild type conditions and what are the binding partners in the mutant. So in that way we can see what is lost and what's gained, and that will differentiate between the loss of function and the gain of function. And we can do that also when we treat with different drugs that activate either the wild type or the mutant, and then be able to interrogate those things. So, and then because 53 is a transcription factor that will induce overexpression of some genes, and then we can see then what are 53 mediated overexpression of particular genes, and then do a kind of a therapeutic characterization of which would be actionable and good therapeutic targets. So that's more to come, and I hope that this has been useful for all of you. And I just want to thank everyone from our group and the different collaborators that have been also part of this work, and also Cookies for Kids, Tim Connor, Leukemia Research Foundation, the Elsa Bardi Foundation, and RAP for funding my work. And with that, I will take any questions. Thank you very much.
Many thanks to our expert speaker, Dr. Diaz-Flores, for his presentation. We are now going to move to the questions submitted by our online viewers. A quick reminder to those watching us live that you can still submit your questions by clicking the Ask a Question tab on the right, typing the question into the message box, and then clicking Submit. So our first question is, from all the different steps you took to increase the virus titer, which one do you think makes the biggest difference?
Great question. So I think the biggest difference was culturing the cells that have been producing the virus for additional 4 to 5 days. So as I said, like the normal protocols that most people use is within the week. So you plate the cells on Monday to have 70% confluence on Tuesday, and then you transfect them and you collect virus at 48 hours. That's the most common protocol. And what we did is, yes, we collect that fraction, but normally now we do it at 72 hours. So that would be on Friday. And then we put more media and then let them grow. And I think what happens is that the cells stop proliferating and then just stop going into production. And then they have been producing virus, so then they keep producing extra virus without growing. So it's just, I think the metabolism changes in become production mode. And then that's when we see the really, really high levels is when we collect the next week on Monday or Tuesday, where we have this really high viral titer. And I think that's the biggest change from all the different things. Even lentiboosts work really well, but I think what makes the biggest difference is that extra culture time, volume, and then concentrating those.
Okay. How would you say the Levy-cell system is helping you with your CRISPR process?
Great. So like we really have been relying more and more on the Levy-cell. So for CRISPR, either if we do nucleofection is basically electroporation, so putting cells into a electric shock. So there's a lot of cell death that occurs. When you do lentivirus production, the polybrin and the other reagents also induce cell death. So what happens is that in the first days, either nucleofection or lentiviral transduction, you have a lot of dead cells. And let's say, just for an example, you get like 50% dead cells and 50% alive. Those 50% dead cells are releasing like dead cytokines that will keep inducing cell death into the other cells. So when we do also puromycin selection, the same. So you go for purifying the transduced fraction. So then puromycin will kill. So if your transduction efficiency was low, let's say that you got 30%, 70% of cells will be dead, and then those cytokines will be killing more of those 30%. So that would greatly reduce the numbers of cells and then extend the time until you get the numbers that you need to collect cells and that you of your interest. With us, we like at the next day after doing any of the nucleofection or transfection or transduction, we use the Levy-cell. So then we start with like highly pure cells, like 95% viability. So then from that day, the cells start growing. It doesn't matter how little you have. So because of that, we have incorporated the Levy-cell to all our CRISPR workflow and also to any of the different work that we use with primate samples and also cells that have been subjected to drugs. So at the end, you are able to like highly purify your cells in a very gentle way, really fast. It's not like a fast sorter, which can be like sorting for an hour or longer per sample. So that's what we have really incorporated in our system, in our workflow, basically.
How important is cell purity and viability in your process?
Really important. The viability, as I mentioned, is one of the things that will really affect the outcome of your results. If you keep dead cells or dying cells, will really reduce the number of cells that you rescue and you harvest. And there's certain downstream application for where that becomes crucial. So for instance, if you are, one of the things that we do a lot is engraft PDX, so patient-derived xenograft mice. So if you put like 1 million cells and you don't know your viability, or you are like expecting that still you transduce cells or you just throw primary cells or whatever, and you just hope that most of them will be alive, that is not the case. And then the engraftment will take much longer. Whereas in our case, we highly purify, and as I show, like really low levels of cells that we need now. and then high viability, so we get better engraftment and faster. And in terms of, so that's with primary sample, but for downstream applications like single cell RNA-seq or like RNA-seq or sequencing, having populations where you have live and dead cells increases the noise. So you really want to avoid noise as much as possible. And then you might have cells that are inducing apoptosis. So then your RNA-seq data will show like some apoptosis induction. And you think that that's part of your, like the question that you are interrogating, but it's not. It's just because you had like a fraction of cells that were dying because of the protocol that you were using. So I think having the purity and the high viability really increases not only the yield of your experiments, but also how clean your experiments are.
Okay, one viewer is asking, with the extended culture time that yielded the high lentiviral titer, do the HEK cells not start to become infected themselves and die and disrupt the viral producing factory?
Great question. No, up to that time, we didn't see any change. The main reason was also that, let's say on Friday, we collected the volume. So all the cells that get stuck are still alive. Otherwise, they would have been removed when you took this initial supernatant. And then, so you put fresh new media, and then the cells do really well until next week. We then try to extend that to see how much can you extend it, but then the yield goes down. So the cells are probably exhausted by then, and then they're like too confluent, and then you lose all the benefit. But I would say like up to the Tuesday of next week, or even Wednesday, you get really high titers without reducing the viability of the cells. After that, your yield will start like dropping significantly.
Do you see better transduction efficiency and editing efficiency in mute P53 or P53 null line than the wild type P53 proficient cells?
Great question. We don't see differences. So that's telling us, this is a great question. This is something that we also consider. Is the way the cells are gonna increase the transduction or not? And surprisingly not. So regardless of the one that has the mutant, the one that have the knockout, we see the same. What really makes the difference is the quantification of the virus. Then we put it and we get the same value. So I think because lentivirus is not depending on proliferation of cells, then like the phenotype of the cells and the behavior of the cells really doesn't affect transduction efficiency.
Have you used the Levitas system to fractionate cells of different phenotypes or just live dead separation?
Great question. We have used it for different phenotypes. So for instance, thanks to the Levy cell, I was able to isolate in these hypodiploid cells some cells that were looking higher. So I have this upper band that I was mentioning, or your live cells. But then on top of that, there was kind of a little other line, less cells. And I was wondering what that was. And I saw it consistently. So then basically I did two-way sort. First, get rid of the dead cells and then rerun them. So then I have more clear separation of that upper band. What it ended up being, that upper band is cells that are much bigger, and ended up being that they reduplicate the chromosome content. So instead of, let's say, these cells can be like 30 chromosomes, so then it's 60 chromosomes. So that in hypodiploid patients is called masked hypodiploidy. And a low hypodiploid patient and a masked low hypodiploid patient, they are treated the same. And it's not clear what makes the difference between having, let's say, a clone that has the 30 chromosomes versus one that has 60, that just reduplicated the entire chromosome. And this is a common occurrence. So what we think is that those have, like, kind of, they evolve, they proliferate less, but they might survive better some treatments. So this is something that we start exploring and it was thanks to be able to do this Levy cell. So that's another kind of phenotypic difference that we did. We have also tried other things, and we have some collaborators who have like done very different cell types, including Drosophila that are very big and they cannot be separated with conventional flow cytometry. You can also separate nuclei. So the nuclei part we're gonna be like doing for like single cell for ATAC-seq experiments that we want to do. So yeah, this allows us to do different phenotypes, and I think we only have scratched the surface in terms of what the capabilities of the Levy cell for the different phenotypic cells can be used for.
How do you think the workflow you showed can be used for interrogating other cancer drivers?
Great question. So I think it can be used for like many. One of the things, for instance, we had been interrogating is the relevance of BCL2. So we published that hypodiploid cells are very sensitive to venetoclax. Thanks to that, the first clinical trial was open for venetoclax in this high-risk population. They do really well, except for those that have the 53 mutations. So the patients that do not do great, the person with like 53, they kind of relapse. So we're interrogating what, so that's one of the work that we are ongoing. But one of the things is what happens to BCL2. Like, so venetoclax can really decrease the viability, but there is some 5-10% of cells that survive. So we basically did a CRISPR of BCL2, and then almost every cell looked dead. So by regular flow cytometry sort, I would have been able to say these look a little bit less dead. So we have to use an inducible CRISPR. But I then put the cells in the Levy cell, and then there was a small fraction, but very clean and clear, of live cells. So we're able to isolate those cells and grow them, and they were like the cells with no BCL2. They just had increased the levels of MCL1. So, and that's why I think this was another way to integrate a different driver in the sense that this was not a mutation. BCL2 is not a mutation, but it was driving the survival in these leukemic cells. So now we are also interrogating for different projects different genes of interest, and we are using the same workflow. So what I think this experimental approach allows you is to interrogate any gene, whether it's relevant for survival or not, because you can always isolate the population, the fraction that is highly viable, and then be able to interrogate those. So I think both, like first the CRISPR part and then the purification part, they can be applied to like any let's say any workflow of people wanting to interrogate anything we have without having the confounding factor of dead cells in your system. And another one, for instance, will be dropout CRISPR screens, where you just put the cells with a drug, and then you have a CRISPR library on them, and then you just wait a week to see what survives, and then you contrast with the day one, and that's where you see, okay, everything that disappears is because it was targeting an essential gene. So in this case, you could be separating those fractions daily, and then know what was the fastest or most relevant gene for survival at day one, at day two, at day three. So multiple things that can be done in this way, I think.
Unfortunately, that's all we have time for right now. It just remains for me to thank today's speaker, Dr. Diaz-Flores from University of California, San Francisco. Please go to the URL now at the bottom of your slide viewer to learn more about resources related to today's discussion, and look out for more webinars from Science, available at webinar.sciencemag.org. If you'd like to receive alerts about future webinars, please sign up using the link in the Resources tab to the right. This webinar will be made available to view again as an on-demand presentation within approximately 48 hours from now. We're interested to know your thoughts of this webinar. Send us an email to the address now up in your slide viewer, webinar@AAAS.org. Again, thank you to our speaker and to Levitas Bio for their kind sponsorship of today's educational seminar.
Thank you also to Science for inviting us to present, and thank you everyone for attending.
Goodbye.
Bye.